Well, on top of the functions, there tends to be classes of proteins. Our purposes, there's going to be two main ones, fibrous and globular. A globular protein is smaller, rounder, and generally plasma soluble, blood soluble. As a result, they tend to be transport proteins more often than not. These are things that are able to float through the blood. Fibrous proteins tend to be much more nonpolar, longer. They tend to be in the cell wall, or at least part of them is in the cell wall. Fibrous proteins are things that give structure more often than not, and so they can connect multiple cells together. They can hold internal parts of cells together, but the idea is they don't really dissolve well in the blood. They don't float through your bloodstream. So they're far more often structural and storage than they are transport or enzymatic. All right, so let's talk about how shape determines which of those functions and which of those classes that you are. So let's imagine I have an alpha helix. And I have nearby another alpha helix. Well, why would these two want to be near each other? There are R groups. And let's say I have a non-polar R group here, and I have a polar R group there. Well, remember the R groups come off of our alpha helixes. So on our other side here, I've got, and if I have another, let's say something very polar, this can hydrogen bond. And if up here I've got isoleucine, I have lots of non-polar interaction. If the R groups happen to line up, that they interact well, my two different alpha helixes can stick to each other. If my R groups were such that they didn't line up well, then I would expect that they would just turn away. So when you have a complex tertiary structure, something like this, those are terrible alpha helixes, but you have some alpha helixes interacting, and you have some beta sheets interacting, or you form a beta sheet. Well, why do those alpha helixes want to interact at all? It's because of these interactions. Now, additionally, remember, they don't just go on inside. There's R groups coming off all sorts of sides. They come out at all angles. And so there's some inside component and there's some outside component. So there's some inside area where some of the R groups can interact with each other on the inside. And there's definitely some outside area where the R groups will interact with whatever's on the outside. So depending on what R groups you have, that alpha helix, so that pair of them, is going to want to be in different places. So let's assume I've got this alpha helix again. So alpha helix down, alpha helix up. Right? And we've got the inside pretty good. We say, hey, here's some non-polar. Use the valine groups. There you go, a bunch of non-polars on the inside. If on the outside I have a bunch of polar, that is, where does this protein want to go? Well, if I've got polar on the outside, that's going to love water. Water's going to interact with it really well. And you can be like, hey, lots of water interaction. Beautiful. That means this is very soluble in water. In fact, you want the inside to be non-polar, because if the inside was polar, it might open up and water get in there. And so by having the inside be non-polar, it's kind of oily, it tries to seal itself up to get away from the water. And the outside being polar loves the water. And so this would dissolve very nicely in the bloodstream or in the cellular fluid. But flip it just a little bit. Same alpha helix idea. Except this time, let's put some really polar things. I've got some O minuses. I've got some N pluses. I've got a lot of ionics in my inside. So my center is super polar. And then on the outside, we'll just put, I like valine, isopropyl groups are easy. Now we have non-polar. I'm doing it short as NP. Non-polar, non-polar. Well, this is not going to like water at all. If water is around the outside, this doesn't want to be there. In fact, it would probably open up and get into the inside and break this structure apart. So, where would this structure be good? Well, this is going to like somewhere that is non-polar around it. Well, we've learned a couple places that are non-polar, but inside the cell, the most non-polar bit you've got is your cell wall. So if I have fats, or phosphodilipids, so O, P, O minus, and here I have, there's a carbon there. Well, if I have my phosphodilipid, non-polar and non-polar, they interact really nicely. And so I would have my fats dissolve this protein pretty darn well. That's actually what we see. If you think about your cell wall, where I've got, I'm just going to shorten my fats as a polar ball with two tails. We talked about the cell wall, the phospholipid bilayer. So we got our polar bit facing the water outside the cell. We got the inside layer facing the water inside the cell. And we've got a bunch of non-polar in the middle. Our protein is going to love hanging out in this non-polar area. So depending on which R groups we use will change whether that molecule wants to be in the bloodstream or whether it wants to be inside the cell wall. This is very important for where we put proteins. You don't want a globular protein meant to transport oxygen sticking in your cell wall. It needs to move. Hemoglobin on your red blood cells needs to keep going. It needs to move around your bloodstream. On the other hand, if you've got connective material holding your cells together, you don't want that to dissolve and drift away. You want it to keep those cells held together. What position our R groups are in? What the R group is at that position changes what it interacts with. Whether it interacts internally well, what type of thing externally it interacts with well. All of those are affected by the R groups. So when we see a very large protein like this, why does that interact? Well, whatever the R groups coming off and facing each other here need to line up. Whatever the R groups facing both those two helixes need to line up. We don't know what they are, but whatever the groups over here are need to line up. And if this is trying to dissolve, in the blood, then all of the outside needs to line up with water. Curious part, if you've got a cell wall though, you can mix them. So you could have a protein that looks like this. And so we'll bring the cell wall over. We would have a very non-polar on the outside edges. We could have very polar holding the insides together. So I'm going to color code it. Let's do blue is polar. And let's do yellow is non-polar. So we'd have some very non-polars on the outsides. We have some polar on the inside. Well, out here where there's water, so in the bloodstream, the outside needs to like water. So there's going to be a bunch of polar on the outside. But how do you get those alpha helices to stick together? Well, if they're in the water, then you would make the inside be the non-polars. The idea is, why would these two parts of the strand stick together instead of letting water wrap around them? Well, because they're non-polar on the inside. And so depending on which R groups you use where, you will favor different domains inside the cell. If the outside of your protein is nonpolar, it fits into the cell wall. If the outside of your protein is polar, it tends to be in the water. Internal components of the protein tend to be opposite so that they will stay stuck together internally. And so those R groups, I mean, there's a reason for them. We need a lot of different ones. They need to be just the right spacing and distance in order to not push each other away. If I have a alpha helix and it has a really long lysine group or whatever, and this alpha helix tries to come up and it runs into it, it doesn't fit. It can't assemble itself. You need the right one for the right distance. It just depends on how that protein is put together. If you ever tried to do any woodworking or anything, you needed to measure down to the 1/8 of the inch or closer to be certain of what you were building. Little differences can cause lots of problem when you're constructing things. And so back when we looked at amino acids, very top, we looked at some of the different ones, and we had cations that were slightly longer and bigger cations, or we had anions that were just an extra carbon in length. We had a bunch of polar bits that were one extra carbon. We had a bunch of nonpolars that were just a few extra carbons in size. That slight adjusting of size is important to make the structure be able to hold together. So, discussion points for this one, big takeaways, kind of the classes, and we start to see where those classes come from. If your outside is very polar, it's easier to be globular. If your outside is very non-polar, it's easier to be fibrous. If you have a mix of the two, depending on how your molecule assembles, well, then you can anchor yourself in the cell wall. And that is actually most proteins. The vast majority of proteins are proteins that actually sit partially or fully in the cell wall, which, curiously enough, is not most of the ones we have structures for. If you've ever seen a large image of a protein, I showed you one with hemoglobin, but if you ever see those kind of structures, most of them are from globular proteins, because if the way you get a protein is you dissolve it and then you slowly evaporate it until it grows a crystal. Crystals we can put to X-ray devices, they can shine light at it, the atoms deflect the X-rays, and we can backwards calculate, based on the deflections, where the atoms were, how far apart they were, what angles they were at. It used to take years to do by hand, but you can do it in about an hour on a computer now. It's a remarkable tool, but you need to be able to dissolve the things to do that. So dissolving something that's supposed to be wedged in the cell wall is a lot harder. So most of the proteins that we really want to know about, we actually don't have structures for. But this is the idea of how the amino acids as side chains as they come off. So all those R groups, the side chains. As they come off the secondary structures, how does that lead to making a tertiary structure? And then how does that tertiary structure favor different environments in the cell?